Okay, it's a pleasure to introduce Ihan, who many of you already know him, so I don't probably need to introduce him, but it's a pleasure still to say a few words. Jihan graduated from my former university and indeed was a master's student of a former PhD student of mine, so we have a special relationship. He's my academic grandson.
Good grandfather.
So he did a double major in chemical engineering and physics at Boğaziçi University in Istanbul, Turkey. Well, I would say the best university in Turkey. I don't know if there are others here who object, but I think it is the best university and one of the best departments. And then he did his master's also in the same department in the area of computational biology already before joining us. Not surprisingly, he did a great job during his studies. He was very well prepared. He already came with with a strong background. And he undertook a challenging project, actually, a joint between the Taylor lab and my lab at the intersection, let's say, between molecular and simulations, an area nobody wants to touch usually. And also, more than that, he also took a difficult problem of understanding dopaminergic signaling. So central nervous system, how things go at the synapse, and also trying to bridge molecular and cellular systems. So it has been a fun journey, I think. But here we are here to hear more about what he has learned, what he has found, and what would be his future recommendations. The good story is that at the end he got a very interesting job at the UPMC. I'm disappointed to see that he's now analyzing sequences to identify biomarkers and cancer cells. So it is, but that's what actually the goal is, to have enough skills, computing and biological skills, to be able to tackle a broad range of research topics. So with that, maybe Jim would like to add anything?
I think you covered it really well.
Okay, so we'll listen to Jihan.
Well, thanks for the introduction. I mean, you set the bar so high. I'm hoping that I can fulfill at least half of it. So I would like to talk about my thesis work on the multi-scale modeling of the neurobiological systems. If I just say, I mean, neurobiological system, it's so broad. I just worked on some special parts of it. But like, first, I would like to start with like why the brain research is important. And then, brain is a 3-pound mass of a tissue, but it's the most complex thing in the universe, and there are a lot of problems related to that needs to be understood. And especially with the increasing life expectancy, the number of neurological disorders are increasing. And then there are more than 1,000 diseases result in hospitalization or loss of productivity. And then, like if we live maybe 100 years ago, it may not be that relevant because we are dealing with polio or maybe some other disease. But right now, with this increased expectancy, we're dealing with the problems. But brain is not helping because brain has the storage capacity which is larger than any of the storage that any human can imagine, and then the size of the connections, the number of connections is up to trillions, which is more than any of the social network that anyone can imagine. Neurobiology has multiple scales. If you go to Dr. Sanowski's website, and then you will see that there's this diagram which is showing that the central nervous system can go up to a scale of 1 meter, whereas some of the transporters or neurotransmitters are only traveling around 1 angstrom range, or maybe a couple tens of angstrom ranges. And then those are very, very different scales, and analysis of those scales we cannot use a single methodology to understand all of them. And also the time scale is varying a lot. Of course, we are not interested right now in this evolutionary scale, but there are a lot of events which are happening less than an hour and then more than a nanosecond range. There are many things are happening over here. we need to bridge different scales to have a comprehensive understanding of the system. And when we're talking about the connections and the bridges, the connections in the central nervous system is between neurons, and they're called the synapses. So basically, they're the junction between neurons. And then the synaptic signaling is one of the main mediators of the brain function. And it also has multiple scales. Because like you see a synapse picture here, and then like in the beginning, you see like, for example, there's an action amplifier. We can travel up to, let's say, micrometers, maybe longer than that. And then like when it reaches to the synaptic cleft, it's just like trigger a neurotransmitter release, and it will just travel maybe let's say 5 nanometer distance, and then like it will trigger the activation of a receptor, which is only, let's say, maybe a couple of nanometers long. So, you know, there are a lot of scales for the synapses also. And basically, I'm more interested in the synapses, and then like you can find different classification of synapses, but for right now, for my purposes, I'm just dividing them based on their neurotransmitter types, and then like one of them is the dopamine synapses, and then like they're generally related to the reward pathways. And another one is the, let's say, the glutamate receptor, glutamate synapses, and then they're more related to memory and learning. Of course, there are like multifunctional synapses with different neurotransmitter types, but like I'm just focusing on one single type. I'd like to come to what is dopamine. Dopamine is a neurotransmitter that belongs to the catecholamine family, and then like there are some hormones, epinephrine and norepinephrine is derived from this dopamine. And more importantly, its function as a neurotransmitter, and then that is discovered by Arvid Carlson. And actually, it ended up with, he got the Nobel Prize in the physiology and medicine because of it in 2000, by his discovery of the relationship between the dopamine and the Parkinson's disease. And, you know, like the dopamine is important because it controls a lot of different things. It controls the motor function, it controls the reward pathways, it controls the addiction, drug abuse, and then like many disorders like the Parkinson's disease, schizophrenia, and ADHD. So basically, this is a dopaminergic synapse in a very, very simplified form. There's like an action potential coming from the presynaptic cell. It triggers the release of this dopamine, a vesicular release of the dopamine to the synaptic cleft. And that released dopamine can activate some of the receptors on the post and presynaptic cell, or it could be cleaned up by these dopamine transporters. Basically, it will reuptake these dopamine molecules inside the cell. But that sort of events are taking place in a very complex environment, because your cells are, especially neurons, are wrinkly, curly, and then like there are very thin channels between them. If we assume that it's a smooth, infinite box that everyone is interacting with each other, I think we are doing something wrong. And then this is a very good example of a synaptic system. This is not a dopamine neuron, but it's a hippocampal an apical dendrite from a hippocampal neuropil. And if you look at this, I mean, it's not that obvious because of the projector, but like you see that it's quite complicated. There are many, many different types of cells in a very, very crowded environment. So we need to use this spatial information in our modeling, and then like when we are doing the computational modeling and when we are trying to learn something from this models. For dopamine system, it was not a part of the models for a long time. I mean, the first dopamine transport model is introduced in 1994, and for like 10 to 14 years, there is no spatial content in this. And there's a slight improvement introduced with the volume transmission model, but which uses only 2 parameters to define all this spatial complexity. I mean, like there are 2 parameters, namely void fraction of the system and the tortuosity of the surface. But there's only one tortuosity for whole tissue. But like it's heterogeneous, it can change a lot. So we introduced a specially realistic multi-scale model of the dopamine neurotransmission. And this is basically our pipeline. This is like a general pipeline for any of this spatial modeling. And we used MCell as our spatial simulator. And then MCell, on the basics, needs 2 things. It needs a reaction network, composed of something. then it needs a geometry. You can define any geometry. It could be a simple geometry, or it could be like a image-based driven geometry. But you can feed any geometry, and MCell will calculate, do this calculation, this simulation, and as a result, it can give you, let's say, some snapshots from the trajectories. You can make some movies out of it. You can see, like, you can make a real-time tracking of each molecules. And also, you can get some information related to those trajectories, like, for example, how, let's say, the activation of one molecule, or, for example, the concentration of a molecule inside a cell. But for our system, we were not that lucky. I mean, when we go to literature, we did not find that much kinetic information about how these dopamine transporters interacting or uptaking these dopamine molecules. So we go and then use some molecular simulations. Colleague, Dr. Mary Cheng, did some molecular simulation over 2 microseconds, which are like very, very long simulations, basically state-of-art simulations. And as a result of it, I mean, she did a lot of work, and then like she identified 4 major states for the dopamine transporter, which is like which are divided based on their gating. It could be outward facing or inward facing, or it could be dopamine bound or dopamine unbound form. And afterwards, she used this rigorous dynamic calculations to get the binding, unbinding rates, and then like the equilibrium ratios of this dopamine transporter. It's not like a 100% or an exact calculation, but those estimates helped us a lot when we are doing the cellular level simulations. So basically, after doing all this molecular work, all this molecular scheme that you see becomes like some arrows and molecules, which is a reaction network. We don't have a geometry of the system, and we use imaging data to reconstruct that. So that Dr. Sorkin's lab is making experiments with the mouse model with hemagglutinin tagged dopamine transporters. And then when they do their imaging study, they can see where the dopamine transporters are located in particular cells. And then specifically Ethan, a lot of imaging in different regions. And we are specifically interested in this striatum region because there are a lot of dopamine transporter in this part. And, for M-cell models, we cannot just go crazy in terms of length scale because we are dealing with diffusion, and then it will be really slow and sometimes intractable to do that sort of simulations. So we are focused in a small region of 10 micron by 10 micron by 7.2 micron, which is already way larger compared to any of the M-cell model that we can find. I mean, for example, there's a model published by Dr. Sinowski's lab, and then I think it's like 10 smaller in terms of volume compared to what we have. And more volume means more molecules, and more molecules means more interactions, and it's slow simulation. So, you know, we are just with a very, very large scale system. And basically, we use this information to understand where the dopamine, the cells that are expressing dopamine transporters. And we made the 3-dimensional reconstruction of the system. And basically, these are our dopamine transporter expressing cells because we have a stack of information. We got a stack of images, and then we made 3D reconstruction from that, and then we got this information. But we can also use this information to identify where are the active zones of this system. We can just like go through the varicosities, where are these blobs, and then from the blobs we can find regions. Like, because we know that active zones doesn't have any dopamine transporters, but there are a lot of dopamine transporters on the surroundings. So we use this information to identify where the dopamine transporters and then, where the active zones, and we find there are 6 of them. But the missing thing in here is that since this is a fluorescent microscopy image, there are other cells around there, but we don't see them because they're not tagged. So in this work, in this 2015 work, the Eaton, Dr. Sorkin's lab published, they find out the distances between those different cells, and we use that information to build the 3-dimension, basically we fill the gap between dopamine transporter expressing cells and other cells. So In the end, we got the geometry and we got the reaction network. So we can use MCell to get some trajectory.